Well, hello, hello. Today we still got potential, but we won't talk about potential. Actually, we will talk about potential. Sorry. We're gonna talk about myelin sheets. Look at this cat. It's so happy under the sheet. Um, actually, that's incorrect. We are actually going to talk about boom, 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 boom. What? Look, this cat, how cute she is. Myelin sheath. Sorry about that joke there. Um, I literally, when I heard the term the first time, I thought it was sheets, like bed sheets. But then I learned I was wrong. It is myelin sheath. And fun fact, sheath is used to refer to the protective layer that covers a knife or something, or a dagger. I'm not sure if dagger has covers. But if you're a samurai and you have a katana, that sheath would be called saya. Now we've digressed. Now, myelin sheath, what it is. So if we look at this picture here, we have two type of neural cells or nerve cells in our system. One is very bare. We're very, very bare, like weathered tree in the winter without myelin, and it sends slow signal. And the other one is myelinated and sends fast signal. And what are the myelins? Those like pinky chunk, like, I don't know, your favorite meat on the barbecue steak. Not sure if this is a good reference. Along the axons here. Remember, we talked about axon in our previous videos. So today we're gonna unpack what is myelin. So remember last video we had a simplistic drawing of a neuron. And you notice that the color is slightly changed, but the concept and the function and the components remains the same. I was just a little bit OCD in the color coordination. But then, okay, let's see. This is a neuron without myelin sheath. And we have our chemical component here. We have, oh, this is too thick. We have our neurotransmitters binding to the receptor, and in turn, internal becomes way more positive. Remember, at the initial stage, at the resting membrane potential, the outside is more positive, the inside is more negative. How negative? Around negative 70 millivolts. Now, we have this gates, right? We call them the voltage gates. We have the green one for sodium, the yellow one for potassium, and when, as we discussed, it is more like a wave when the action potentials travel and not a straight line. And what that means is that, hey, we've reached here. Here is a little bit more negative, and here's positive, and the positive was attracted towards the negative, reached the threshold, about minus 55 millivolts, fire. What does it mean is that when it fires, this gate actually opens, right? And the positive sodium rushes in, makes this even more positive, and then we continue to the next section, we continue to the next section, we continue to the next section. But this is gonna be a slow process, right? So if we imagine we're driving a car, this is a very cool car, by the way, and we have encountered some road barriers or the gate. Actually, this is a very vivid illustration of how the voltage gate opens. It kind of just opens like this. We open the gate, boom. We close the gate, boom. And Imagine every time when you drive, this is gate 1, okay, can you open it? Are you ready to open? Open. You reach gate 2, are you ready to open? Not yet, wait a little bit, okay, I'm open. Then you can go to 3 and 4 and 5 and 6 and 7 and 8. And this is a very, 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 very slow process, right? We call this process, this whole process of action potential travel through different patches or sections or like continuously, it's called continuous conduction, right? Continuous conduction. It just refers to this process here. What about with myelin sheath, right? With myelin sheath, similar things, but you can see we've got some buff here if you're a gamer. So we still got our neurotransmitter here. This one becomes increasingly positive. And what will happen? First, you notice that there are much fewer gates here. And there's something here that we call myelin sheaths in the pink here. And now, when this one becomes overly positive, same thing happens, right? Initially, inside the membrane is negative, outside is positive, and we reach the threshold, we fire, and this one becomes positive, right? Inside becomes way more positive, and we're gonna attract to the next one. But this is actually a misleading drawing. We are not gonna jump. It may look like you're jumping. Sorry, not you, but action potential is jumping. Just look at this little shoot rod here. It's jumping, it's jumping, it's jumping. Oh, we're not jumping, right? So what do we do if we are not jumping? What happens is that we actually just travel through the next section. And we appeared again. And the same thing happened. We increase the positive charges inside the membrane. Bam, 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 bam. Positive threshold hits. We fire again. And then you won't see this, but it's going to travel to the next gate. Or not gate, to be honest. This gap here is called a node of Ranvier. Or if you want to pronounce it in English way, it would be node of Ranvier. So it may appear that the action potentials are jumping from node to node, but that's actually a misconception. Just because you don't see it flowing doesn't mean that it doesn't go through the myelin sheaths. Now, how do we understand myelin sheaths? It's actually very interesting. So imagine the same road is here, and we have gate 1, gate 2, gate 3, right? And now, imagine you are driving the same car on the same road, but only this time there's mountains in between, and you have to drive through, not drive above, I think that's a suicidal mission, drive through the tunnels to the next node, to the next node. Now, what makes it fast? Well, there are two ways to make this fast that, one, if you look up, we have much more gates in here. Much, much, much more. We have like 8. Here we only have 3. So we reduce the waiting time, right? So that's number 1 to make it fast. Number 2 is that imagine if you understand the basics of physics. So if you go through the tunnel inside the mountain, it's actually an isolated environment. There's less wind resistance. So you can accelerate through the tunnel and then appear again in gate 2. And just because we don't see you traveling through the tunnel because of the mountain blocked our view, it doesn't mean you're jumping from 1 to 2 to 3. We can accurately say that you appear to jump from 1 to 2 to 3. All of these nodes. And myelin sheaths, this is how myelin sheaths work. It accelerates the action potentials traveling. And another thing is, see, there is no, bulletproof. Sorry, that may be a wrong reference, but every gate as tight as it may be, some gate will leak, right? With the myelin sheaths, there's no gate. No gate means no leak. Theoretically, no gates, no leaks. And we can save all of the action potentials, the positive areas, and push them forward and, fire the signal much faster. And this is why neurobiology is so interesting. Now, this process is called saltatory conduction, right? Saltatory conduction. And my mistake, I thought is something related with salt, but actually no. The word saltatory comes from Latin saltare, meaning to leap or to jump. But remember, we're not jumping. We just appear to be jumping. And that's how the myelin sheath signal travels faster than the neurons without myelin sheaths. Now, where does myelin sheath come from? Put it simply, we'll have a whole other video talking about nervous system. And for the moment, let's look at it in a very simplified way. We have our nervous system. And the nervous system consists of peripheral nervous system and central nervous system. And then in the peripheral, wait, shorten as PNS, please. It's not PNS. There is a difference. It's PNS. That contains Schwann cells. By the way, it's Schwann. Apparently, it's a German guy, not the Swan. As much as I like it to be, it is not. And for the central nervous system, CNS, contains, wait for it, oligodendrocytes. And they are both a type of glial cells. I think there is an I somewhere. Anyways, and glial cells is responsible or produce the myelin sheaths. But bear in mind that not all glial cells are involved in myelination. Myelination is a word to refer to the process of myelin production. For example, astrocytes are not responsible for myelination, but we'll have a later video focused on glial cells specifically because I think these little guys are very interesting as well. So that is where the myelin sheaths come from. So one question when I was learning about myelin sheaths was that why do we have both? Why can't we just have, the neurons with myelin sheaths? They are much faster and it's much more efficient and productive. No. If we think ourselves from an evolutionary perspective and more like a, investor, there are certain costs for, fast signaling. Number one is a biological cost. We need much higher energy. Now I want a burger. We need a much higher energy cost. Because the myelin must be built, maintained, and repaired. And hence, if you look at the damage, if your myelin sheaths are damaged, it requires repair, and diseases such as multiple sclerosis demonstrate that maintenance can be a burden. So the damage to myelin sheaths, there is a proper term for it. This one is it. It's called demyelination. And so it requires energy, it requires constant maintain, build, and repair. Hence, we have high biological costs. And then there's also structural costs. Everybody nowadays want to have low body fats, look good for the selfies on Instagram. But if we have all of the neurons with myelin sheaths, we basically will be very bloated. We'll take up more space because it's much thicker. If you don't believe it, just look at this photo again. Skinny, thick, right? So what else? What else? What else? What else can we say? Functional costs. Sorry. Also, larger nervous system require fewer neurons that can fit into the same volume. We don't want to be bloated, so we're gonna fit a little bit less neurons. Functional costs. Many signals don't need high-speed digest. Like, for example, digestion, gut mobility, temperature sensing. Like, your brain doesn't need to know or doesn't want to know, like, hey, my temperature is currently the 36 degrees Celsius, and in next minute it's like, okay, 36.1 degrees Celsius. We don't need that level of detail, so hence some neurons are not with Myelin sheath. And here is a quick table, just some examples. By the way, when I say, I try to word it very accurately here. When we say heavily myelinated, it doesn't mean that there is 0 neurons without Myelin sheath. I always say unmyelinated, there might be a slight chance that there is 1 or 2 stuck in there that with Myelin sheath. I know, human body, very exciting. So, reflexes. Say, you're sitting on a table, the doctor knocked on your knee, your reflex. That's good. You're playing tennis, and there's a volley. You hit it. That's nice. That is very high needle for speed, and hence it is heavily myelinated. Voluntary movement. Hi. Imagine there is a spider on your back. You're gonna shake it off, right? Like Taylor Swift. Flame touch, very high. Shock pain, yes. Remember when you touched the stove and it was like, ouch, and you immediately put your hand back? That was medium to high, thinly myelinated. Dull pain, low. Sometimes I'm glad that there are unmyelinated pain because it is slow and we feel the pain a little bit less. Anyways, digestion, low. There are many unmyelinated fibers, and organ regulation, low to moderate, and is a little bit mixed. And that's it. That's why we need 2 types of neurons. In summary, today we learned about bad sheath. Sorry, we learned about Myelin sheath. We learned about continuous conduction. We learned about saltatory conduction. We learned about why we need 2 types of neurons, and we also learn about where does Myelin sheath come from. So here's a nice summary table. We have our Myelin sheaths, and which means we have myelin covering, and we have supporting cells, and we have Schwann cell and oligodendrocytes. We have signal transmission, and action travels continuously into the entire membrane for the unmyelinated neuron, and for the myelinated neuron regenerate only at the nodes of Ranvier. Conduction type continues, and saltatory, remember, it has nothing to do with salt, it's more to do with the jump. Speed, unmyelinated slower, myelinated much faster. Energy efficiency, it is lower. Myelinated neurons higher. Current flow, depolarization occurs at every segment of the axon. As we mentioned, wave after wave after wave. And with myelinated neuron, it is current travels roughly beneath the myelin between the nodes, like we're traveling through the tunnel inside of the mountain. That's it for today's video. If you like it, please give me a like, comment, or subscribe. If you have any comments or feedback, please write to me down below. And until then, I will see you next time.